Good morning, afternoon, or evening, wherever you are. My name is Athena Lemon, and I'm the project coordinator for the Wolbachia Project. This might be a little bit different than a normal research in progress seminar because the Wolbachia Project is a science outreach program. As you've heard in previous talks during the seminar series, Wolbachia is an obligate intracellular endosymbiont discovered in 1924. Hurtig and Wolbach noted that this type of organism is distributed through the whole group of arthropods, and their observation is held true. Out of approximately 1 million named arthropod species, and however many more there are that are unnamed, mathematical modeling estimates that about 50% of the Earth's arthropods can be or are infected with Wolbachia. This makes it ideal for educational use. Arthropods, bugs, are abundant and easily collectible. Furthermore, as previously mentioned, Wolbachia is a master manipulator and is a reproductive parasite. It has a host of really interesting phenotypes. Students are drawn to charismatic arthropods like roly-polies and ladybugs, and awed by the stories of how cytoplasmic incompatibility reduces mosquito populations, and interested in using Wolbachia for vector control. Now that we have a refresher on what Wolbachia is, let me introduce you to the Wolbachia Project. The Wolbachia Project started about 16 years ago at the Marine Biological Lab in Woods Hole, Massachusetts. It is a collaboration between scientists, educators, and research specialists. Eventually, Seth and Sarah Bordenstein moved to Vanderbilt, and the project followed them about 13 years ago. The project is now housed in the Bordenstein Lab at Vanderbilt University, where I joined the program recently, and is the outreach arm for the Bordenstein Lab, whose goal is to understand and disseminate principles that shape interactions between animals, microbes, and viruses. So what are the goals of this project? We want to encourage students to become scientists. We ask students to engage with the world around them, participate in discovery and problem-based learning, and encourage them to participate in the scientific community. In turn, we affect the community the students are a part of. We are able to engage the community with a proven scientific outreach program that encourages scientific literacy, facilitates collaboration with research scientists to describe arthropod biodiversity. There are millions of species of arthropods and only so many Wolbachia labs to characterize the Wolbachia and arthropod diversity across the globe. What is the workflow of the project, and how does this actually work? First, students learn about biodiversity. What can they find in their area? They hypothesize what may not have Wolbachia, and when students go out and actually collect arthropods, they are able to test a hypothesis. Then they practice biotechnology by macerating their bug, macerating their arthropod, and extracting DNA. Then they amplify an arthropod barcoding gene and a 16S Wolbachia DNA by PCR, and visualize those fragments by gel electrophoresis. Finally, students learn about DNA sequencing and phylogeny, and then have an opportunity for their arthropod and Wolbachia DNA to be sequenced. Throughout this process, they're adding data to our new Wolbachia Project database. This database, created by Dr. Nick Negretti, is a new shared repository for Wolbachia Project data. This is a picture of 2 student scientists in Cincinnati a few weeks ago. They're collecting and testing periodical cicadas for Wolbachia. I'm actually getting the first 25 sequences from them this week. We are bringing common molecular biology techniques and real science to the high school classroom. Before our database, any data that they found would either stay in their classroom or likely not escape beyond their school district unless they published. We wanted to create a database to be able to collect all of the data that was being generated in disparate classrooms into a central location. There are 3 main groups that will benefit from the database. First, students and citizen scientists are able to submit independent research to be published in the database. Each entry has a unique code, so they're able to put the published post on their CV or resume. And we've set up the database like a lab notebook, so it's something good to be published, to be seen. Secondly, students really want to contribute real, meaningful data. Their eyes light up when they learn that they can help researchers and do real science and contribute to the symbiotic community, the symbiosis community. Speaking of the symbiosis community, we were able to track arthropod biodiversity, track Wolbachia frequency and distribution, and track Wolbachia sequence and arthropod sequence diversity throughout all the samples that are collected. Like I mentioned earlier, there are only so many Wolbachia labs in the world. Having excited, motivated student communities study not only biodiversity, but sequence diversity and Wolbachia infection rates across the world is a project no single lab could take on itself. Finally, educators will find the database extremely useful. Each lab or scientist, we all have our own favorite kits, and they all might work differently depending on whose lab you're in. In the classroom, the kits work differently between each classroom. By collecting the methods that students extract DNA, run their PCR, or stain their gel, we can determine what method is most effective for classroom use. We've had to use word of mouth to troubleshoot problems. For example, Sarah Bordenstein, the director of the Wolbachia Project, was able to determine that a popular post stain for gels was not good enough for detecting the occasional low titers of Wolbachia. Each classroom is its own lab with different experimental processes, and the high school laboratory is different from a research laboratory. We can test multiple kits out in the world in the classroom setting and recommend protocols accordingly. Also, for educators, we're able to facilitate partnerships with scientists. An educator in Atlanta, Georgia was having difficulty extracting DNA from roly-polies, so we connected him with Richard Cordeau, who is an expert in the field. Through his mentorship, his class in Atlanta, Georgia now rears locally collected isopod colonies for Wolbachia studies. Let me show you what the database looks like. This is our home page of the database, and by going up to database and then browse, you can actually look at all of the recent posts that have been submitted to the database. And this is just a fun way to kind of get an idea of what's on the database. You can also notice there's a tag W+ or W- to quickly see if an arthropod is Wolbachia positive or not. You can also search by map, by location, and here we're zooming in to Nashville, Tennessee, which is where we're located. And by clicking any one of these dots, we can go directly to the post that it's associated with. We can also search by taxonomy, which is something that I really like and I think is really cool. We have over 150 arthropods, over 150 hexapods, in the database right now. Let's say that I have a really specific question and I only want to look for Wolbachia positive arthropods. I can do that with our advanced search function, and I can click on this and go directly to a student entry. This is actually a student entry, and this is where she caught this roly-poly, and it was wild caught, and there's some more information on that database post. Let's say we're going to submit some data. I would click on the gold box called Submit Data in the top right-hand corner of the database and start filling out my new post. I would name it something. Let's say that I found a lady beetle and fill out my observations. It's really easy to put in a location because you can click and drag the map. So for example, if I was in Liverpool, England, and I just found this lady beetle at a park, that's what I would put in. And I would go down the observations list. As I did the project, I would update my methods and finally fill out my results. This can be saved, published, or previewed at any point. And over the course of the project, you can go back and save your draft or continually just publish it and update your published post as students go through the project. As researchers, we want to know, can we trust the data? Can we use this data? And our answers to that are participants are encouraged to upload images of gels and assess the confidence level of their Wolbachia identification. So this is actually built into the results section where they put in a confidence level. And again, this is a real post by a high school student where this high school student says that she's very confident that it does not have Wolbachia because she interpreted the controls correctly, and explains how she interpreted that. The database page is set up like a lab notebook. The back end and front end, when you look at it, looks like a lab notebook. There are observations, methods, and results. And all of the methods and results are readily available to the scientific community. Gel images are embedded in the database page. Here's actually a picture of a gel from those two students that are looking at cicadas, periodical cicadas. And this is a real picture of a gel that they are uploading to the database. This is a duplex PCR, so this top band is the arthropod COI, the arthropod barcoding gene, whereas this bottom band is the Wolbachia fragment. And you can see they don't have a lot of Wolbachia, which is hypothesis based. That's kind of what they expected. Although this result looks really interesting. I digress. We also want to know if, as researchers, if we can use the data. All data can be exported as a CSV file. I'll show you how easy it is right here. You just click the CSV file and it automatically downloads. And it's really easy. All of the sequence data is freely available, and any pictures that the students or participants take are available with attribution to the contributor. And there's a little spot when you upload your photos for photo credit. In addition, students or researchers can blast directly from the database. So you're going to watch me blast the arthropod CO1 sequence from this database post at NCBI. So there I am clicking the NCBI, opening up NCBI for me, and while that's running, I can also blast it against the sequences in our database. We only have about 250 entries right now, so this particular entry, which is a black carpenter ant, is matching the other sequenced ants in our database, the rust-colored carpenter ant. I'd also like to tell you some really cool stories of how the Wolbachia Project has affected students across the globe. There's an international collaboration between Christine Gertain in Toms River, New Jersey, and Dr. Pirshi Waxman in Israel. And they have a really cool collaboration where their students go out and collect similar arthropods on 2 different continents. And their students then get together and talk. They have conferences and discuss what they found and compare results and findings. They had a little bit of a hard time last year due to the pandemic, but they still met over Zoom to discuss their findings, which is really cool. In addition, there's novel data discovered due to the Wolbachia Project. So students in Switzerland first identified Wolbachia in a local fly species. It had never been identified in that fly species before, and they're continuing to expand their biodiversity studies. And those same students in Israel also identified Wolbachia in a desert beetle, which was previously unreported. And they're now collaborating with a local university to further the research. Both of these examples are examples where Wolbachia has never been found in that arthropod collected. So how can you or a local high school teacher join the Wolbachia Project? You can, of course, always contact us at our email, which is here, wolbachiaproject@vanderbilt.edu. We have all of our free curriculum and resources online on our website, vu.edu/Wolbachia. These are all at the end of the presentation. And you can attend office hours, which is where I open up a Zoom link for an hour and a half every other week. And you can ask me anything. You can ask me questions or troubleshoot and just get in touch with me that way. You can always receive at no cost PCR primers and controls. These are insect controls and DNA controls for your classroom. And then you're able to facilitate student-driven research and upload the results to the Wolbachia Project database, which is what I had demoed previously. So what are some opportunities for collaboration? You can run the Wolbachia Project at your home institution. We've had the Wolbachia Project integrated into both undergraduate and high school curriculum, and we're happy to do that. The Wolbachia Project is free to use, free to share. And you can also use it as an outreach opportunity to your local community. One more point about the undergraduate and high school curriculum. Introduction to Biology or Independent Student Research are great classes for this, for the Wolbachia Project, because it's an introduction to molecular biology. You can also, as a researcher, submit data to the database. We have a spot on the entry form in the observations where you can put down if it's a wild-caught arthropod or lab-raised arthropod. And so you can actually submit data. You can go through the Wolbachia Project in your own lab, maybe with your own samples, and take pictures, have a geographical location, and upload that data to the database to give students a more research-heavy database or research entries to look at. And finally, even if you're not interested in this, even if you're not a Wolbachia researcher, we invite you to participate in Scientist Saturday on social media. We're interested in showing students this is what a scientist looks like. This is what we look like outside of the lab coats and what we do outside of the lab. So we have started a Symbiosis Scientist Saturday, in which we feature a scientist, and you go through a questionnaire, answer some questions, and we are always happy to feature symbiosis scientists. So if you're interested in that, please email us, and we can start that process off, get some pictures, get some information from you, and then have you featured on our social media accounts. And so with that, thank you very much for your time and attention today. And I would just like to go through a couple of these links. The first link is our website, vu.edu/Wolbachia, where we have links to the curriculum and any additional information you might want. Our social media handles for everything is Wolbachia Proj. Our database that we've been looking through is wolbachiaprojectdb.org. And finally, our email, feel free to email us with anything, wolbachiaproject@vanderbilt.edu. I am having office hours tomorrow, 6/16, and office hours are from 3 to 4:30 p.m. CST, and you can email us for that Zoom link, and I'd be happy to see you there. Thank you so much for your time today. Have a great day.